Okay. The Committee on Science, Space, and Technology will again come to order. Without objection, the chairs are authorized to declare recesses of the committee at any time. Good morning, and welcome to today's hearing entitled The Science of Zika, the DNA of an Epidemic. Let me recognize myself for 5 minutes for an opening statement. Our second witness today is Dr. Daniel Neafsey, Associate Director, Genomic Center for Infectious Disease at the Broad Institute of MIT and Harvard. In this role, Dr. Neafsey leads a research group studying the DNA of mosquito vectors and the pathogens they transmit. He has developed pioneering resources and computational methodologies for genetic studies of mosquitoes and mosquito-borne pathogens. Dr. Neafsey received his BA from Loyola University in Chicago and his PhD from Harvard University.
So, Mr. Chairman, Ranking Member Johnson, and members of the committee, thank you for holding this hearing and for the opportunity to present today. I also want to acknowledge one of the committee members, Representative Clark, an important supporter of the strong research community we have in Cambridge, Mass. My research group at the Broad Institute in Cambridge uses DNA sequencing to understand and control infectious diseases, and I'm here today to speak specifically about how DNA sequencing can help us to understand and inform the control of Zika transmission. We lack fundamental understanding of how the Zika virus moves from person to person via Aedes mosquitoes, information that is crucial for an effective response to this epidemic. Mosquitoes are not flying hypodermic needles that passively transmit disease. The successful transmission of mosquito-borne disease depends on complex biological and ecological interactions between mosquitoes, their human hosts, and the pathogens that they transmit. Insight into the biology of how Aedes mosquitoes are able to spread disease can be gained through sequencing and mapping the Aedes genome, or the entirety of its DNA. We know that because we have demonstrated this with Anopheles mosquitoes, which transmit malaria. So I led a project at the Broad Institute recently, funded by the NIH and involving more than 130 collaborators from 19 countries around the world, to build genome assemblies or maps of all of the DNA of 16 different kinds of Anopheles mosquitoes, some of them very proficient at transmitting malaria and some of them deficient. We learned a lot of things from this project, but if we can advance the slide, I will show just one small result. This is a figure that we built by comparing the genome maps of these different mosquitoes and identifying those groups of genes that were changing most quickly or most slowly between these different mosquitoes. As you can see, the most rapidly changing genes at the top of this list in red are those genes that we know to influence how mosquitoes taste and smell. And we know that there are important genes in this class that determine whether mosquitoes prefer to bite people or animals. And a strong preference for biting humans is a very important determinant of disease transmission. So close behind those smell and taste genes are genes that provide mosquitoes with an immune system. And we know that variation in these genes can determine whether or not mosquitoes are able to transmit pathogens or control infections and not transmit them to the next person they bite. So no comparable set of genomic resources yet exists for Aedes mosquitoes, despite the fact that they transmit Zika, as well as the 3 other major viral diseases that Dr. Ernst mentioned. Next slide, please. Here's an image showing the current state of a recent Aedes aegypti mosquito genome map. The map is in approximately 3,700 pieces, despite the fact that this mosquito has only 3 chromosomes. So the target is 3. The original map for Aedes mosquitoes, published in 2007, was in about 10 times as many pieces, so we're making progress. So as mosquito season begins in the Gulf states, and with Zika on the doorstep of the United States, we lack foundational resources to pursue DNA-based studies of the biology and transmission of Zika. This resource gap is critical. Infectious disease epidemiology has been transformed by DNA during the last 10 years into a rich digital information science, allowing biologists and public health agencies to track the spread of outbreaks over time and space and learn about what mosquito and human factors contribute to disease spread. We can now tackle emergent infectious diseases like Zika using efficient and innovative genetic tools to build databases and situational awareness of diseases. And the scientific community stands ready to develop and apply these tools to the Zika epidemic to protect vulnerable populations within our borders and around the world. One example of that readiness is the Aedes Genome Working Group, Organized just this last January via Twitter by Dr. Leslie Vosshall at Rockefeller University, this upstart group's mission has been to produce an improved genome map for Aedes mosquitoes using new sequencing technology that is 10,000 times cheaper than the technology available 10 years ago. With no central funding, but a lot of donated time and resources from a range of academic and industry partners, just motivated to improve our capacity to respond to Zika, this group has made rapid progress exploring a spectrum of new technologies for building an improved genome map. So with continued support and follow-up research, this map will yield knowledge that informs disease control measures. Examples of such measures include insecticide resistance detection and surveillance, mosquito population studies, and genetic modification of the mosquito. New tools for which are creating an increasingly direct connection between basic DNA research and control measures. The Zika epidemic can become a proving ground for the power of new DNA-based resources and epidemiological tools. Taking advantage of rapidly falling costs, we can demonstrate to the world how new technologies will let us understand, anticipate, and control the spread of an epidemic, and we have an obligation to vulnerable populations to seize this opportunity. So thank you to the Chairman, Ranking Member Johnson, and the committee for your attention, and I'll be happy to answer any questions you may have later in the hearing.
Thank you, Dr. Neafsey and Dr. Presley.
Sure. I think this question probably falls closer to the expertise of my co-panelists, but I think it would be reasonable to advise caution, especially for pregnant women who might consider traveling to a Zika endemic area. Okay. But to echo what Dr. Ernst mentioned, beyond geographic considerations and altitude, there are also seasonal dynamics that surely contribute to Zika transmission. So it's
what would be the most dangerous time of the year to travel?
Oh, I think the hot, wet time of year.
Summer?
Yes, but I think at this stage it's very difficult to quantify exactly what that risk represents.
Okay, thank you. Second.
Neafsey, on the mapping, how close are we? Or how far are we, depending on how you look at it?
I think we're close. I think we're making good progress towards having a better genetic map, but I think having a better genetic map for the mosquito is a proximal goal. And the goal of applying that map most fruitfully to studies, for example, of insecticide resistance and identifying those markers that are most useful in the field for maintaining the efficacy of the control measures we do have, that will take further application of the genome map and testing of more samples.
Thank you. And
right. So I second the the importance of surveillance, and I can speak to work being done by one of my colleagues at the Broad Institute, Pardis Sabeti, who was well positioned to apply DNA-based studies of the Ebola outbreak because she was establishing a research base in West Africa and starting to characterize fevers of unknown origin. There are many agents of infectious disease that result in symptoms and that are undiagnosable because we don't know what the agent is. And there are many of these, particularly in tropical parts of the world, any one of which could, in principle, emerge to cause an epidemic. So I would advocate the importance of surveillance, the importance of catching new epidemics early, and the utility of DNA-based studies for getting an early characterization of perhaps the agent behind emerging outbreaks.
To the panel, I've just got a couple of science questions. Is this a intracellular or an extracellular virus? Does it live in the RBC, WC, or is it in the plasma? Where does it live in the human?
I think it replicates in cells, and I think work is underway on elaborating exactly which cell types are most favored by the virus. I think there was this convincing work a few weeks ago with brain tissue organoids to establish its affinity for developing neuronal cells. But I would say that the full preferences for the types of cells infected by the Zika virus are still being elaborated upon.
And Dr. Ernst, you said.
Dr. Ernst, in your testimony, you identified some of the knowledge gap areas about the interaction between the Zika virus and the mosquito that spreads it. I was also curious, Dr. Neafsey, the work you're doing to sequence and map the genome, is that going to help fill in some of the gaps in knowledge, and kind of where are we in that process, timelines, those kinds of things?
Sure. So I think I can attest to the value that genome sequences can have as a resource for informing precisely the kinds of parameters that make the types of models Dr. Ernst described accurate. In the malaria field, we've been using this kind of data to inform models of malaria transmission and malaria distribution. One example of the way these data can be used is a study that was published last year by investigators at Notre Dame who did genetic profiling of Aedes mosquitoes from the Capitol Hill neighborhood and used the genetic signature of these mosquito samples over the course of several years to determine that this was a year-round population, likely a year-round population, taking refuge in sewers or below ground during the cold winter months, rather than a new population that was being refounded by genetically different sources with every successive warm season. So this is a small example of some of the resolution that DNA-based data can lend to some of the parameters that are useful for understanding the existing and maybe the prospective distribution of mosquitoes.
And I was just reading a little bit about, you're kind of communicating via Twitter and periodically getting together? Are there like kind of work projects in place, and how do you monitor who's doing what? And
yeah, so Twitter is a fantastic scientific resource. I don't know if it's recognized outside of the scientific community, but news travels so quickly. It is unparalleled for organizing people who are like-minded and who want to get behind a common cause. So Twitter, I think, and a lament about the quality of the existing Aedes mosquito genome map was the genesis of this working group I alluded to. Since then, we've had phone conversations, emails, but as yet no in-person meeting. As I mentioned, this is a kind of a volunteer group, and we've been working catch as catch can to this point.
I know you don't want to commit to a timetable of when, the completion of the assembly of the DNA map, but can you give us a rough ballpark?
I can say that we have parallel efforts underway using several different new technologies that we all hope will yield a better genome map on the order of a month or two. Okay. These are unproven, but we are very optimistic given their performance with sequencing human genomes and assembling maps for human DNA. So I think we're optimistic that we won't have a perfect genome map by this fall, but we will have a map that is significantly improved beyond the existing one.
Okay, thank you very much. I yield back.
We appreciate the information that you all are bringing to us today. I wanted to start with something very specific and sort of move to the more general. The impact of this virus on a fetus is one of the most frightening aspects of a rapidly changing landscape. But do we know now exactly how the virus causes birth defects like microcephaly, and do we have an understanding of how exposure at different times of pregnancy can impact the fetus? Or is this a gap in our research?
I don't think we have a virologist on the panel, but I'm going to step slightly outside my comfort zone just to provide a basic answer and say that the general principles that determine what cells and what hosts a virus can infect often come down to the particular proteins or other attributes that are on the surface of those cells. The virus usually needs to recognize a doorway and be able to interact with it in a way that permits entry. So some of the work that's been done to date has taken advantage of some of these fantastic new resources in the laboratory, such as these brain organoids, very simple, very small, three-dimensional tissue replicas of what a developing brain might look like. And the realization that has led to is that there are particular cells at stages of brain development that are indeed susceptible to Zika infection. I think there's much work to be done to understand, as I mentioned previously, the spectrum of cell types that could be susceptible to Zika infection and the precise proteins and sort of molecular interplay that's responsible for Zika's ability to infect the cell types and the range of organisms that it does infect. on the host side as well as, let me say, the mosquito side. Its ability to proliferate within mosquitoes and be injected into people that are bitten subsequently is another aspect of the molecular biology that I think we need to determine.
Yeah, Dr. Presley.
If I could add, ma'am.
Is the better way. One of the most terrifying conversations I ever had was at the Broad looking at antibiotic resistance and infections. So how can we do a better job of funding basic science that allows you to do the applied science that can bring us some answers in 22 seconds?
I can speak briefly. I think what's important is to set up an infrastructure for collecting and pooling information. I think there are a lot of investigators collecting diverse forms of information, and I think there are opportunities to really rapidly advance our state of knowledge about Zika and other emerging epidemics on the horizon by organizing that information, creating communication channels, not just exclusively via Twitter, but other media, and making sure that we are performing surveillance, that we're collecting the kind of information we need to, in the short term, limit the advance of these diseases, and in the longer term, understand them so that we can develop longer term control measures.
Great. Thank you. I see I'm out of time. I yield back.